Could you let me know when you get the message?
It's streaming now.
Okay, so I fix that. Now I share. Okay, now we're good to go. So, welcome everyone, and thanks for joining our seminar, our fine seminar today. I am Eliana Maldonado Chaparro. I'm professor at Universidad del Rosario in Colombia, and I'm really excited to host this seminar today. Before I keep going, I want to thank Matthew Silk for his seminar on how demography and disease shape animal social network dynamics. It was a great seminar. And looking forward to next week's seminar, Lauren O'Connell, shifting gears on our study system, where she's going to be talking about family dynamics in a biparental amphibian. So this one has been also been on the list of things to watch for a lot of people. And just a couple reminders about our seminar. It's going to be about 50 minutes of talk, followed by 1 hour of discussion, 30 to 60 minutes of discussion. Just make sure to keep your microphones muted. And then at the end of the talk, we're going to have time for discussion. So please make sure to type your question mark, and I will call your name. And if you're a student, just make sure to type the S next to it, and we'll give you priority. For those who are on YouTube, please type in your question, and Lauren will help us relay it to the speaker. So with no further ado, our speaker for this week is Dr. Sylvia Kaiser. Sylvia got her PhD at University of Münster in 1997, and she is currently an adjunct professor at the Department of Behavioral Ecology at the same university, where she has also been working since 1998 in different roles as professor and research assistant. Well, Sylvia's research covered different aspects of social behavior. She's mostly interested in development of social behavior, and her focus is prenatal phase and adolescence. And she works or look at these questions from the hormonal and molecular perspective. She also works on cognition, emotion, and on stress and stress avoidance. And she has contributed a lot to animal welfare. She's actually a member of the university's animal experimentation committee, where she has contributed with a remarkable work. As far as her publication record, I mean, it's impressive. One of the papers you read for today, the last piece on in specialization and social niche, I think it's really, really exciting. And with no further ado, I'm just going to let Sylvia talk. Her seminar title is Modulation of Behavioral Profiles by the Social Environment During Sensitive Phases of Life. Sylvia, I'm going to stop my share, and then you can go ahead.
Yeah. Thank you very much for this really very kind introduction. I'm very happy to be here and to give this talk here. I will also share my screen. So I hope you can see the presentation. It's a correct mode also? Yes. Okay, good, great. So I have to put this here so I can now also see my slides. Okay, I would like to speak about the modulation of behavioral profiles by the social environment during sensitive phases of life. And I think all of you are very well known that individuals can differ in their behavior, not only the appearance or physiology or so on, but also the behavior is differing between individuals. So they can be aggressive, they can be less aggressive, they can be shy, they can be bold, and so on. Of course, genes plays an important role in the developing these differences, but I will not go into that in this talk. But of course, genes play the role. We know that genes can control the behavior. They don't determine the behavior, that's very important, but they are involved in the controlling of behavior. But what I wanted to show to you today is that there is another very important key factor in modulating behavioral profiles, and that's the environment. So there are a lot of different environmental factors which can be involved in these shaping processes. That's abiotic factors, as for example, temperature or other climate factors. There are also biotic factors, and here we can distinguish between heterospecific factors. For example, predators plays a very important role, but also the conspecific. So the social world, the social environment in which the individual is living can have a large impact on the development of the behavior. And this is that we are focusing in our research. We wanted to know, to understand which role does the conspecifics play in behavioral development. There are several very important phases during ontogeny, during which environmental factors can act on an individual and so shape the behavioral profile. And I would like to focus here on mammals because we are working with mammals, but of course also in other systematic groups, there are such phases and also environmental factors can shape the development. But here I would like to focus on the mammals, and I would like to show to you here 3 phases during which environmental factors can have such shaping processes. The first is the prenatal phase. That's a very important phase in mammals. So not only with birth, these developmental processes starts already during pregnancy. Of course, environmental factors can have a shaping process. But the environmental factors cannot act directly at the individual because there is a kind of protection in the womb. But the environmental factors can act on the mother. And because then there are specific physiological, for example, physiological reactions, these factors can be also affect the individual in a way that this individual shows then later in life as adult individual a specific behavioral profile. I will show to you that later. But the idea is that during the prenatal phase, environmental factors act on the pregnant organism, and this has effects on the individual, so there is a specific behavioral profile later in life. The next very important phase is the early postnatal phase. That means in mammals, the lactation period, also a very important phase. And this phase is characterized by a more or less strong attachment to the parents, particularly to the mother, because the mother has the milk, of course, and it depends on species how big the role of the father is. There can also be, of course, a lot of parental care coming from the father, and how strong the bonding is between mother and father. It depends on species. But there is an influence of the mother and of the father on the individual. And here we see that environmental factors can act on the one hand directly at the individual, at the lactating, at the non-weaned individual. But again, it can also act indirectly on the mother and then mediated by the milk, for example, by hormones in the milk or by the maternal behavior, can also then shape the behavioral profile of an individual. I will also show you an example in a minute. So we see that in this early postnatal phase, during lactation period, environmental factors can act on the individual, and this leads to a shaping process that there is a specific behavioral profile later in life. And the last very important phase I would like to show to you is adolescence. That's also a very important phase because in this phase, the individual will become more and more independent from the parents, but the peers will have more and more influence. So they play now an important role in the life of the individual. Of course, dispersal can occur during this phase of life. Depends also on species and also on the sex, whether dispersal occurs. And very important, the individual will become sexually mature. So they will now receive the ability to reproduce. And here, the environmental factors can act again on the individual, in this case only directly. And this has then again a shaping process, so that a specific behavioral profile will develop, which is then seen in the adult individual. So we are interested in all these three phases, and I would like to show to you how environmental factors can shape the profile, the behavioral profile in the individuals during these three phases. And our model species is the guinea pig. So that's a domestic species. The wild ancestor of the guinea pigs are the wild cavy. They are living in South America. They are very common there. We are at the, I will also show some studies regarding the wild cavies, but I would like to start with the guinea pigs with a domestic form. And we have chosen the guinea pigs because we are interested in the role of the conspecifics on the social environment. And guinea pigs are social animals. And for this kind of questions, it makes sense to use social animals and to investigate this kind of research. So what we have done is the following. We had pregnant females living in two different social environments, a stable or an unstable social environment. And that does mean that the following, so all animals lived in groups with one adult male and 5 females, as you can see here. And part of this group composition was always the same. So we never changed the group composition in these groups. So that means that the animals were living in a stable social environment. So they have their conspecifics. We don't change it. So several months they were living in the same group. The other animals were living in an unstable social environment, and that means that we have changed the group composition in a regular way. That means that we have exchanged 2 females every 3rd day, so that the group composition was constant for not more than 3 successive days. And because in each group 5 females were living, the same female was transferred every 15 days. And pregnancy lasts about 68 days in guinea pigs, so that's a long time for such small mammals. But that means that we could transfer each female also several times during pregnancy. Because of practical reasons, we have then in this study, we have then the females were living also in the same social environments during lactation. So that means that also the lactating females were living in a stable or an unstable social environment. In this study, we cannot distinguish between the effects of the pregnancy or lactation period. So that means that the lactating female was transferred together with her pup in a new enclosure when the female should be transferred. So that means the pup was then living also during lactation in these different social environments. And with the age of 20 days, that's the time of weaning in guinea pigs, we put them out of these groups and put them now into offspring groups. That means each 2 females or 2 sons were put together. And the 2 animals of one group were coming from the same social environments, or a stable or an unstable social environment. And from this time on, we didn't change the group composition, so the offspring was then all living in a stable social environment. And now we wanted to know what are the effects of these different early social environments, so the stable or the unstable social environment, on the development of the offspring. And I would like to show to you first the effects on the daughters. And we could show that daughters of mothers who have lived in an unstable social environment during pregnancy and lactation showed a behavioral masculinization. So that means they showed behavioral patterns which are normally shown only by male guinea pigs in mixed-sex groups. So, for example, the so-called rumba, that's a male typical courtship behavior in guinea pigs. You can see here a female and here a male, and the male is dancing around the female. There is also a characteristic vocalization. I hope you can hear it. And that's a really male typical courtship behavior. In mixed sex groups, only males are showing this behavior. In female groups, also females show this behavior, but lower frequencies than males, but you can see that. But interestingly, when we are comparing our daughter groups, then we see that the daughters whose mothers had lived in an unstable social environment show this behavioral pattern at a level like males are doing that, and significantly higher in higher frequencies than the females whose mothers had lived in a stable social environment. And another example is the intensive nasoanal sniffing. You see here again a male and here a female, and the male is licking and sniffing intensively at the anal region. So we see there is a behavioral masculinization in daughters whose mothers had lived in such an unstable social environment. And interestingly, this behavioral masculinization correlated also with higher testosterone concentrations. Testosterone is the male sex hormone, of course, and the main source of testosterone are the testes. Of course, the females have no testes, but nevertheless, each female mammal has also testosterone because there are also other sources, as for example, the ovaries or also the adrenal glands. So females have testosterone, but at a lower concentration than males. That was also the case here. But what we could show that was that the daughters whose mothers had lived in an unstable social environment, here the red part, showed significantly higher testosterone concentrations than the daughters whose mothers had lived in a stable social environment. The concentrations were lower than in males, but they were significantly higher than in the daughters coming from this stable social environment. So we see there was a behavioral masculinization and also a masculinization of, let's say, the endocrine system, because we also find these increased testosterone concentrations. Okay, that were the daughters. What's going on with the sons? And here we found also differences, but in a different way. Here we could show that sons whose mothers had lived in an unstable social environment during pregnancy and lactation showed a so-called, or we have called it, a behavioral infantilization. That means they showed a delayed development. They showed behavioral patterns which are normally shown only by very young male guinea pigs, for example, resting with bodily contact. No adult male will rest with bodily contact with conspecifics, but young males do that. And here we see they do that until an older age than normally. And also some play behavior, they are also shown more frequently and up to an older age. So that's a behavioral infantilization. And this was interestingly also combined by a delayed development of the pituitary adrenocortical system. That's one of the main stress systems in mammals or invertebrates. And a good indicator for this system are glucocorticoids. The main glucocorticoid in guinea pigs is cortisol, the same as in humans. That's a little bit strange because guinea pigs, of course, are also rodents, and the most rodents have corticosterone as a main glucocorticoid. But in guinea pigs, it's cortisol. And we know that there is a typical course with age of cortisol concentrations, indicating this developmental stage of this pituitary adrenocortical system. And we could show that the sons whose mothers had lived in this unstable social environment showed a delayed development. That means they reached the adult levels up to first in an older age than the other sons are doing that. So what we could show was that in guinea pigs, these early unstable social environments can have sex-specific effects on the behavioral development. And now we were interested in two different questions. The first was, what are the underlying mechanisms behind that? And so I would like to explain this with help of this figure. So we have an individual, a specific individual with a specific behavioral profile, for example, masculinization, infantilization, or non-masculinization, or whatever. Of course, what I have told to you at the beginning of this talk, genes can play a role in the development of the behavioral profile. I will not go into that. But what I want to emphasize here is that the early social environment in which the pregnant and lactating female is living can shape the behavioral profile. And the question is, how can this work? What's the reason behind it? What's the mechanism behind it? And one main factor which is discussed is our maternal hormones. So maternal hormones can be can shape the phenotype of the offspring in a specific way. And how can this work? Often it is discussed in the following way. So that, for example, the unstable social environment in which the pregnant or also the lactating female was living represent a stressor. And of course, if there is a stressor, then the organism is recognizing that, and then physiological stress reaction will occur. For example, stress hormones will increase. So, for example, as I have told you, glucocorticoids as cortisol, but also other stress hormones as, for example, catecholamines, epinephrine, norepinephrine are also very important stress hormones, will be also released in stress situations. And interestingly, also androgens can be influenced by a stressor because, as I have already told to you, a stressor will activate the pituitary adrenocortical system, and glucocorticoids are secreted because of this activation. But androgens are also secreted in the adrenal glands, so also in the cortex of the adrenal gland. And so if there is an activation, then also the secretion of androgens can be influenced. That's the idea. And that means that the hormones which will be then secreted because of the stressor will circulate in the maternal bloodstream. And at least partly it could be shown that these hormones can cross the placenta and reach the fetus. And so it can then have effects on the differentiation, also on the sexual differentiation of the fetal CNS. That is the idea, and in a lot of species that could be shown that this is also the case, and we also wanted to know, of course, whether this is also the case in our guinea pigs. And there were some surprise, I have to say, because it's not so easy as we sometimes think. So the first what we have done was that we have, for example, applicated ACTH. ACTH is a hormone which is secreted by the pituitary and is activating glucocorticoids. So we have pregnant females living in the same environment, so the social environment was not changed, but we have applicated them with ACTH, and then we thought that there should be also a masculinization effect, but that was not the case. We have differences in the behavior, but not in the maternal behavior. In the masculinization. And then we wanted to know, okay, that's strange, but what's going on regarding what are really the effects of an unstable social environment on the pregnant females? And so we have investigated these pregnant females more in detail. And the first very interesting and really surprising effect was that we didn't find any differences in cortisol concentrations. Even when we have put one pregnant female, put it in a new group, there was no increase of cortisol. So they could cope with this situation very well. There was also no aggressive behavior between the new group members. So they lived very good in this situation. But we found differences in the behavior in another way. So we found that the pregnant females living in this unstable social environment showed a behavioral activation. And maybe we have no data to that, but our behavioral data point to an activation of the sympathetico-adrenomedullary system, the second very important stress system. That means that maybe catecholamines can be increased. We have no data about that, but that could be the case. And there are some studies which say that maybe catecholamines can have a masculinization effect. So that's really very theoretical, but maybe this can be the case. Interestingly, we found differences in the androgen concentrations. Here we have investigated not testosterone, but dihydroepiandrosterone and its sulfate. That's one of the main androgens in female mammals. but it was the other way around, we thought, because we found that these androgens are lower in pregnant females living in an unstable social environment. I have to say, I was pretty sure that we will find an increase because of the masculinization, but we found a decrease. So still we have not really a good idea regarding the masculinization, but we can explain the infantilization of the sons because of this decrease of the androgens, because we have now conducted a further experiment that was a PhD study of Dirk Wevers. And what we have done, we have applicated pregnant females with an androgen receptor blocker, with flutamide. So that means that the androgen concentrations were the same, but the receptor was blocked so that the androgens cannot work anymore. And when we have done that during pregnancy, then we could show that there is again an infantilization of the behavior of the sons. So we have compared in this study sons of mothers who have received this androgen receptor blocker, or with sons who have of mothers who have received only a placebo. And these we found that there is exactly the same behavioral infantilization as we see in our sons whose mothers had lived in an unstable social environment. So maternal hormones can play a very important role in mediating these effects. There is another factor which is also often discussed, and that's the maternal behavior. Of course, this is true for the lactating period. And so we also investigated in our guinea pigs whether maternal behavior maybe is changed due to these different social environments. It is known, for example, in rats and also in mice, that if, for example, there are adverse conditions, then mothers show only a low amount of maternal behavior. So the amount of maternal behavior and also quality of maternal behavior depends on environmental conditions. We didn't find any differences in our guinea pigs, but that was not really surprising because guinea pigs are precocal species. So they are more or less fully developed when they are born. So they have fur, they can see, they can hear, they even eat already solid food 1 hour after birth. So they are really don't need the mother a lot. Of course, they need the milk, but the mother don't show a lot of maternal behavior. So probably that's not plays such an important role. And for example, the sexual differentiation phase is in the prenatal phase and not in the lactation period. That's maybe also a very important point. In rats and mice, for example, that's different because these are altricial animals. So that means when they are born, they have no fur, they need thermoregulation provided by the mother, they cannot see, they cannot not hear, and so on. So they need the parents really much more than guinea pigs. And here also the sexual differentiation phase, for example, is also in the early postnatal phase, so around birth. So probably here the shaping process is much more important during the early postnatal phase. And there are a study of Mike Meany, a very famous study, in which they could show that if rats whose mothers have shown a high amount of maternal behavior, that these rats are later in life less anxious and less stress reactive than rats whose mothers has received them, provided them only with a low amount of maternal behavior. So these studies show that maternal behavior can shape the behavioral profile. Not in our system, but in other systems, this is the case. So probably maternal hormones and maternal behavior plays a very important role regarding the mechanisms of these shaping processes. There is a second question we are very interested in, and that is whether these different behavioral profiles could represent adaptations. So could it be that the mother can prepare her offspring in an optimal way so that the offspring, a masculinized daughter and infantilized son, is prepared in an optimal way, for example, to an unstable social environment. That's the idea behind that. And this hypothesis is called as an external predictive adaptive response. That's discussed a lot in different studies. And the idea is the following. So if of course, there is a specific environment during early life. So, for example, during the pregnancy, during lactation period, and so on. There is one important assumption, and that means that this environment during early life should predict the environment during adulthood. So if the probability that there is a change in the environment, then this hypothesis makes no sense. But if the probability is relatively high that the environment will be the same when the individual will become adult, then this hypothesis makes sense. The second assumption is that the environment should provide reliable cues which can be recognized by the mother or by the individual. So that means that the individual will then develop based on these cues in a specific way. So that now a behavioral profile or also an endocrine profile will be developed, which represent an adaptation to that environment, so that the individual is prepared in an optimal way to this environment. That's the idea of this external predictive adaptive response hypothesis. Okay, and we wanted to know whether this could maybe be the case in our guinea pigs. And for that, that means we wanted to know whether these masculinization and infantilization could represent adaptive maternal effects. To investigate this, we changed now to our species. So the species is the same, of course, but we go now to the wild caves, the wild ancestor of the domestic guinea pig. That were studies of Katja Siegler in her PhD thesis. And the first was that Katja could show that also in the wild caves, the different social environments during early life has the same effect. So the daughters are masculinized, the sons were infantilized when the mother had lived in a stable or an unstable social environment. So the effect was the same. That was, of course, very important, because then it's clear that these effects of an unstable social environment is not an artifact of domestication or so. That's really general rule, let's say. So we also find it in the wild animals. And then, and then we wanted to know whether these different profiles could represent adaptations to the specific environments. And the idea was the following. So you can see here wild caves in South America in this natural habitat. We have done our studies, I have to say, in our lab in Münster. We had to that time also wild caves in Münster. Here you can see such wild caves in South America. Here they forage and they eat grass, so they need such grass habitats, but they need also structures to hide, such bushes or such things, because each predator is loving wild caves. So they need the possibility to hide. And what it is very interesting, which is common also in other small mammals, is that the population density can change from year to year. So sometimes we have a low population density, sometimes a high population density. And that's the idea what we had. So we think that that a main characteristic of a low population density is a high degree of social stability. So that means our experimental design of a stable social environment represents more or less a low population density situation. Of course, that's more differences, but one main characteristic is this high degree of social stability. And in a high population density situation, this is characterized by a higher degree of social instability. So our unstable social environment mimics such a situation of a high population density situation. That's the idea behind that. And I would like to focus here on males. We would then predict that in a low population density situation, the non-infantilized sons should have advantages over the infantilized sons, should be better adapted. Whereas in a high population density situation, we would predict that the infantilized sons should be better adapted than the non-infantilized sons. So that was the idea behind that. And why should this be the case? So we have these two different behavioral profiles. I have shown to you that this means this infantilized profile and the non-infantilized profile. In a high population density situation, that means that the unstable social, mimic more or less this unstable social environment we had in our experimental designs, we have seen that the sons showed this delayed development. So they develop more slowly, they show this juvenile behavioral profile. And the idea or this behavioral profile, this juvenile behavioral profile should have an advantage in a situation when adult males are present, because males are intolerant to each other. That means if there are two males meeting each other, they will be very aggressive to each other. Such a young male is not strong enough to compete successfully with older, stronger males. And so it makes sense that this male show a so-called queuing strategy, so that this male should wait until it is strong enough to compete successfully with older males before he try to mate with a female. And in a high population density, the chance, the probability to meet an older and adult male is relatively high. And if the male show not such a juvenile profile, then the risk to be involved in aggressive encounter with this older male is relatively high, and then the risk to be injured is also relatively high. And so the idea is, if they show this juvenile behavior profile, they avoid to be involved in aggressive encounters, and so they avoid to be injured, and they have time to grow up and later to mate with females. In a low population density situation, there is another situation. And here we think it makes sense to develop, not in this way I have described with the unstable sons, but that they show now a non-infantilized behavioral profile. And here we could show that they are a little bit more socially interactive. They show a more social orientation and also a little bit more aggressive behavior. And this makes sense in a situation in which no adult male is present. And in a low population density situation, this could occur. Of course, there could also be the probability that there could be also the situation in which the young male will meet such an adult male, and then it will be involved in aggressive encounters, but it is a trade-off. The risk is thus much lower than in a low population density situation, and the probability to meet a female with whom he can mate is also lower. And so in this case, it makes probably sense to show an early reproduction strategies. That means to mate a female as soon as he meets such a female, but to hope, let's say, that it will not meet an older male, so that there will be then an aggressive encounter. So that was the idea behind that. I'm sorry. And that we wanted to prove. And I would like to show to you the example how we have done that to prove whether the queuing strategy could be adaptive. And for that, we have conducted so-called mismatch experiments. What does that mean? So we had in Münster such outdoor enclosures. Such an enclosure was 10 square meters large, and it was connected also with an indoor enclosure, which was 3 square meters large. And in this enclosure, there were also food and water, of course, available, a lot of different enrichment stuff. That's here a symbol for that. We have shelters. The animals were also transponded, so we have also some antennae in these enclosures. In this enclosure, a resident harem was living. That means an older male and 2 adult females. They were living there for several months. And then we have introduced to these harems simultaneously each one young stable and 1 young unstable son. That means a son whose mothers had lived in a stable or in an unstable social environment during pregnancy and lactation. And this means that this situation now represents a match for the unstable son, because he was prepared for a situation that an adult male is present, and this is the case here. And it represents a mismatch for the stable son because he was prepared for a situation that no adult male should be present, but now he is there. So, and now we wanted to know, our prediction was that the unstable sons should have advantages over the stable sons in this situation. Of course, we have measured paternities, but here I have to say that the young sons were not successful, so all offspring could be sired by the resident male. But that's normal also in the wild. We know there were also studies of Matthias Ascher, supervised by Norbert Sachse. Norbert, probably a lot of you know him. He's still senior professor at our institute. And they could show that in nature, multiple paternities can occur. So if the litter is more than one pup, then there are multiple paternities to a degree of 20 to 30%. But they could show that the father is either the resident male, so living in the harem, or it is a male of a neighboring harem, also an older adult male. In a specific situation, there are also young males, which are called satellite males, but they were never able to reproduce in these studies. So probably this is the same. So they need to become older to really be successfully to reproduce. But nevertheless, we have also measured fitness proxies. And here we, for example, have measured cortisol. The experiment lasts 40 days. And you can see here now the cortisol concentrations during that experiment from day 0 until day 40. And you can see we have taken blood samples for each animal shortly before start of the experiment. And so these are the initial values. There was no difference between the two groups of males. The black dots are the males whose mothers had lived in an unstable social environment, and the white bar dots are the males whose mothers had lived in a stable social environment. Then we have put the males in this harem and measured 2 hours later again cortisol concentrations. And what you can see here, also not surprising, there is a sharp increase of cortisol. There was also no difference between these two groups of males. But then we have taken blood samples that after 5 days. We didn't want to disturb too often, so we have then the first blood sample again taken 5 days after introduction into these groups. And we see that the males coming from the unstable social environment have reached initial values. So there was no difference anymore between initial values and these values until the end of the experiment. Whereas the stable sons also show a decrease, but it's still higher than the initial values, and they remain higher until the end of the experiment. So this seems to show us that the unstable sons could cope with this situation in a better way than the stable sons. And so this is an indication that in male wild cavies, an adaptive shaping of the phenotype by environmental influences during early phases of life can occur. So that were the studies regarding a summary of studies of our studies about the early phases of life. In the last part of the talk, I would like to show to you now also that also the adolescence is a very important phase in which such shaping processes can occur. A long time, the focus was of research was here in the early phases of life, but since the last 10 years or so, it becomes more and more clear how important the adolescence is in this context. So we also have conducted studies in the adolescence, again now with guinea pigs. There were studies I have conducted together with Norbert Sachse. And what we have done is the following. So we had adolescent males living again in 2 different social environments. So in such a large mixed sex colony, there are 8 to 10 males, 10 to 12 females and the offspring, and in such a heterosexual pair. So only 1 male, 1 female were living together. And then we wanted to know whether they differ regarding their behavior and their endocrine system. And that was the case. So what we could show was the following. So if we are comparing these males living during adolescence in these two different social environments, then we see that the colony housed males showed a lower level of aggression towards strange conspecific, whereas the pair housed males showed a high level of aggression towards strange conspecifics. And we found also differences in hormones. So the colony housed males showed high testosterone, whereas the pair housed males showed only low testosterone concentrations. And we found differences regarding cortisol responsiveness. The colony housed males showed only a low cortisol reactivity, whereas the pair housed males showed a high cortisol reactivity. We have in our lab standardized tests to measure cortisol reactivity. That means that we take the individual out from the home enclosure and put it for several hours into a new enclosure alone, in which only food and water is available. And if we are doing that, then you can see here a very typical pattern of cortisol response. So you can see at the x-axis the time points at which we are taking blood samples. So before start of the experiment, that's a 0. Then after 1, 2, and 4 hours. At the y-axis, you can see the cortisol concentrations, and then you can see this is the initial value, that after 1 hour there is an increase of cortisol, a significant increase. After 2 hours there is still an increase, and after 4 hours initial values are reached again. That's a very typical pattern of the cortisol reactivity in females and in males, and we see this pattern also, for example, for young males living in these different social environments, a colony or in the pair host males. But interestingly, there is a social modulation of this cortisol responsiveness during adolescence, because if now the adolescent males are living, or if the males are living during adolescence in a colony, we see a different pattern. When they were living during adolescence in this colony, then, and we do conduct this cortisol responsive test, then during late adolescence, so after sexual maturity, but not really fully adult, then we see that there is still an increase after 1 hour, but this is significantly lower than the increase here when they were younger or adult. So we see there is a social modulation of cortisol reactivity. And we wanted to understand this more in detail, and for that it was a PhD thesis of Stephanie Lürzel. We have compared these different housing systems more in detail. And what Steffi could show was first that a very important difference between these different housing situations is the number of social interactions in which the males are involved. So if they were living during adolescence in a colony, so in a high individual number situation, they are involved in a high number of social interactions. There are a lot of different social interaction partners available, of course. And this was very low in the heterosexual pair, because there is only the female with whom he could interact. And what Steffi could show experimentally, that the number of social interactions could shape the testosterone concentration. So the high testosterone concentrations are due to the high number of social interactions, and the low testosterone concentrations were due to the low number of social interactions in the pair situation. The next question was then whether could it be that the different testosterone concentrations could shape cortisol reactivity. So we found in the colony this low cortisol responsiveness, and in the pair house situation, this high cortisol responsiveness. And we wanted to know whether testosterone can organize this cortisol responsiveness. And to investigate this, of course, the perfect experiment for that is to gonectomize animals, because then they have only a low amount of testosterone. And so Steffi has gonectomized colony housed males before sexual maturity. So what she has done is the following: male guinea pigs will become sexually mature with the age of 70 days. and she has gonectomized them with the age of 40 days, so well before sexual maturity. And here you can see now the increase of cortisol concentrations after 1 hour of our cortisol reactivity test in animals before surgery, so with the age of 39 days. And here is no difference at all in the increase of cortisol. With the age of 40 days, she has gonectomized the animals. So these are now the gonectomized animals, and the control group were sham aberrated, of course. And after the the surgery, you can see now that the animals, they are all living in colonies, but now these animals have not anymore high testosterone concentrations, and now they show a higher increase of cortisol after 1 hour than the sham aberrated males, as males living in colonies. And so that's typical pattern like in the pair-housed males we found in the pair-housed males. So the testosterone are really involved in this shaping process. And Steffi has also conducted the other experiment. She has applied testosterone to pair-housed males. And again, you can see here the increase of cortisol after 1 hour. And before start of the experiment, there was no difference between the animals. And then she has applied the animals with testosterone or with a vehicle. And you can see if the males were applied with testosterone, then after a while, the cortisol reactivity was decreased. So they show now a pattern as usually colony-housed males are showing. So we see application during that time leads to a decrease of cortisol responsiveness. So we could show that testosterone can modulate cortisol responsiveness, and there are also some indication that cortisol responsiveness is then responsible for the difference in the aggressive behavior, which we could find in our colony in pair-housed males. So that means that the aggressive the colony-housed males show a low aggressive phenotype, whereas the pair-housed males show a high aggressive phenotype. So what we could show is that in guinea pigs, the endocrine and behavioral profile can be shaped by the social environment experienced during adolescence. And again, as in the early part of my talk, we were also interested whether this difference in the behavioral profile can represent adaptations to the specific environmental conditions. So I'm very sorry. So the idea is that not only the early life, so prenatal phase or the early postnatal phase, can predict the environment and provide reliable cues based on that the individual can develop, but that this is also possible during adolescence. So that means that the environment during adolescence can predict the environment during adulthood. The individual can recognize cues. Developed on these cues, a specific behavioral profile can be developed, and this represent adaptations, adjustments to this environment. So that means that the low aggressive phenotype represents an adaptation to the colony house situation, and the high aggressive phenotype an adaptation to the pair house situation. And again, to investigate this, we conducted match-mismatch experiments. That was a PhD study of Tobias Zimmermann. So that means that we have these two different social environments during adolescence, the pair and the colony house situation. And then Toby has provided a new enclosure for these animals. So he has constructed again a situation in which only a low number of individuals are available, so that are 2 females. And then he has introduced simultaneously each one pair house and 1 colony housed male to these 2 females. So this is now a low number situation, and that means that should represent a match for the pair housed males and a mismatch for the colony housed males, because the pair housed males are prepared for such a situation in which only a low number of individuals there, in contrast to the colony housed males. And now we wanted to know, have, in this case, the pair housed males an advantage over the colony housed males? And we could show, yes, that was the case. So for example, the pair-host males were more likely to become dominant in this situation. And furthermore, the pair-host males had a higher reproductive success than the colony host males. And so we could show that in male guinea pigs, an adaptive shaping of the phenotype by environmental influences during adolescence can occur. Okay, so I'm at the end of my talk. I would like to conclude. I hope that I could show to you that the prenatal, early postnatal and adolescent phases can probably be considered sensitive phases, that the environmental stimuli experienced during the sensitive phases can influence the behavior in adulthood, that an external predictive adaptive response can occur. That means that environmental input provides information about the future environment, and that social interactions during adolescence trigger neuroendocrine processes that shape the individual's behavior profiles and adapt them to the social environment in which they live. And lastly, that the social modulation of behavior profile from the prenatal phase through adolescence represents an effective mechanism for repeated and rapid adaptation. And with that, I would like to thank you very much for your attention. I would like to thank my cooperation partners, especially Norbert, Toby, and Mike, but also, of course, a lot of other people who were involved in these studies. And of course, I would also like to thank my funding, mainly of the DFG. And again, thanks a lot for your attention. Thank you.
Thank you, Sylvia, for a great talk. I think we learned a lot about instability and the effects of it. Okay, so now we open our discussion time, and I'm gonna keep an eye on the chat. So just remember, if you have a question, type in a question mark. I see you are coming, Eduardo. And just remember to introduce yourself and just give your affiliation so we create a sense of community.
Thank you, Adriana. Thank you, Sylvia. I'm Eduardo Fernandez Duque at Yale University in the US. I have a couple of questions, but I'll start with the very first one that goes back early in the talk. I'm very curious, a couple of things. Can you tell us a little bit what makes what are things you measure for defining the situation as unstable? I think you got to that later in the talk when you're talking about social interactions, but I was curious about the first experiment when you set it up, the females, and unstable, stable. What do you mean by unstable? And then I apologize if I missed it, but did you consider the relatedness among any or all of the subjects that you are evaluating, beginning with that first study? I mean, how the beauty of working with captive animals, I usually get very jealous of all the things that you can do, including giving them receptors, blocking receptors, etc. But and you can also probably have an awful lot of information about the pedigree, the relatedness between them, and to what extent that could be one more factor to ponder in assessing the responses you've measured. And I'm thinking about the first study. I have some other questions after others have asked, but.
So regarding, you mean, I'm not completely sure. The first part, you wanted to know what stable means. Or so we have these two different social environments, and stable environment in the first study means that we don't change the group composition.
No, that I understood, but what is it about the change? The change is just a proxy for something else. And what I was curious, maybe that's what, when you, towards the end, you were talking about the number of social interactions. For that first study, what is it that the change brings that it's perceived as stability?
So the idea was that in, for example, in a high population density situation, there is a higher degree of social instability because individuals are dispersed, other individuals are immigrating, and so there are changing individual conspecifics in a group. So that was the idea, so that we have changed the group composition. So the interaction partner, where the group the density was the same in this situation. So there were always 5 females and 1 male, but the interaction partner changed because there was a new female there. And we have seen that then there was a behavioral activation. They showed more social interactions, they showed more locomotory activation and so on. And that's during the whole time. Not only we have introduced every third day a new female, of course, then the behavioral activation, for example, was the highest, but also the other following days, we found this general activation, let's say. That was the main point.
Right.
And for that, I think that probably we have no data about that, but I could imagine that really also catecholamines can be increased because locomotory activity is also correlated with a higher epinephrine concentrations, for example. But we have no data about that. That would, but could be the case maybe. Is this answering your answer? Your question?
Yes.
Okay.
That's what I suspected, that it is something
yeah.
about the characteristics of the social interactions that, that you also
yeah, exactly.
Thank you.
And we also know, for example, I was really surprised that we didn't find any difference in the cortisol concentrations, because we know in female guinea pigs, that's different in wild cavies, because in wild cavies, females don't interact a lot with each others, but in domestic guinea pigs, they also build bonding. So they also build friendships, let's say. And so I thought, if there is a disruption of the group structure, that should be stressful. And of course, we have this activation and so on, but nothing at all regarding cortisol. That was really surprising, because they really interact a lot with, they have favorite interaction partners. And this we have interrupt. So it was a little bit surprising for us.
And since you mentioned cortisol, then I'll take. I see that. I'll ask another question. You're getting blood samples, right, from the captive animals. How long does it take you? What's the timing of handling? How do you control for the peak that you get of an activation of the axis just from the process of handling that has nothing to do with the social context?
So entering the room or the enclosure, in the outdoor enclosures, we have 3 minutes time. And we have shown in guinea pigs that 3-minute time does mean that there is really no handling effect. That's really then the initial values. That's, of course, no problem in our guinea pigs in our indoor enclosure. It was not so easy in the outdoor enclosure, but we could manage it. So it was possible to do that within 3 minutes. And I have to add that we only need 5 microliter plasma. And we take it from the ear vessels. And that is really very fast to do that. And
so you don't go into the vein, it's just a prick from the ear.
Yeah, exactly. And that's enough. And it works very well. Of course, we can also take saliva samples, but that takes sometimes longer to have enough saliva. And the most stressful thing for guinea pigs is not taking the blood sample, but really the handling. And the blood sample is so fast, that's really no problem, but the handling is a problem for the animals, and that we have to do anyway. So that's, but 3 minutes, that's a very important point. We have to be finished within 3 minutes, and then everything is fine.
Thanks.
And then your other question regarding the relatedness of the animals. We have not measured it, but because we of course, the animals were coming from different parent groups, but we breed ourselves. We try to breed as broad as possible, but because we also determine paternities, and for that we need also it's not good if they are the degree of inbreeding is too high, then we have a problem. So this is not the case, but of course there is a kind of relatedness between the animals, but we were aware that in the groups that they were coming always from different breeding groups. So that was the case, but nevertheless there is a kind of inbreeding in our guinea pigs.
Thank you.
Thank you, Dordo. Okay, I have a question from Maren.
Hello, my name is Maren Hook. I'm at the University of Derby. And my question is either to be answered very briefly or probably not. Because one of my favorite stories to tell the students, to tell my students, is about how the Münster yellow-toothed cavy was discovered compared to the yellow-toothed cavy, and the yellow-toothed cavy has if I remember correctly, the same structure as the domestic guinea pig and the wild guinea pig, and the Münster is monogamous. So I was wondering whether you did the same or similar studies also with any of the so either the normal yellow-toothed cavy or the Münster yellow-toothed cavy.
No, because I'm not sure whether everybody is knowing the Münster cavy, probably not. So that was an interesting story. We had the yellow-toothed cavies, the Gallia musteloides in our institute, and there was a high degree of inbreeding. And so we had a guest then there, and coming from Bolivia, and he said, it's no problem. And his ranch, he has a lot of these animals, and he can send some animals to our lab. And he has done that. And then we were it was very strange for us because they look very similar, but they behave in a much different way. And because the yellow-toothed cavies, the Gallia musteloides, they can be kept in large groups. They show also a promiscuous mating system. They could be aggressive to each other, but they can manage that very well. And we know also that there is, for example, a high degree of multiple paternities, for example, and so. And the thing was, when we have received the new animals, you cannot keep them in large groups because they were monogamous and they were really intolerant to each other. And when we wanted to put them together, we tried at first with pairs. It was also not so easy to establish new pairs. So a male or a female, we have to try which animal are like each others and which not. And so it was not so easy. And for that, we cannot conduct such studies with an unstable social environment. They will kill each others. And that's a problem. And it's very interesting because these species are really very relative to each others, but they have much different mating and social systems. So the Gallia monasteriensis, that we have then investigated them much more in detail, especially Norbert. And we could show together with the Senckenberg Museum that it is a new species. And then we said, what's the name? And we have said it's a Münstersche guinea pig, so Gallia monasteriensis, that's coming from Münster. And we have conducted several studies, but the interesting thing was really that they behave in a much different way. And so we have not really conducted this kind of experiments that would not be possible in this species.
And in the normal yellow-toothed cavy, were you able to do something similar with them? Is it similar?
No, with them, we have only done these studies with the wild Cavia aparea, so the ancestor of the guinea pig and the domestic guinea pig. The problem was still with the Gallia musteloides, we had this high degree of inbreeding and we had no new animals. And so we had not enough animals for this kind of studies. And so we have to stop to work with the Gallia musteloides. That was the problem with them. So we have no data about that.
Thank you, Maren. And do we have a question from Lauren?
Yeah, thank you. And Sylvia, thank you for that talk. It was really fascinating. My name is Lauren Hayes. I'm a professor at the University of Tennessee at Chattanooga, and I study social degus in Chile. Of course, I'm familiar with this work, and I enjoyed hearing it all put together. I had a question, and it kind of follows up on Eduardo's questions about composition. in these experiments where you manipulate composition, do you know anything about the extent to which transfers of females disrupt sort of the extent of how similar phenotypically the adults are? So I'm getting at the context of homophily of adults that we've been exploring in the Degus, and I just wondered if you could speak to that.
I'm not completely sure whether I understood the question. What exactly do you mean?
So in the Degus, we know that, you know, similar phenotype females based on anal distance is important to fitness and
yeah, sure.
And I'm wondering if your groups, you had any index of that.
Yeah, you mean, I hope I get it now.
Oh, it's okay.
So you mean that when the animals are adult, whether then they, what's the composition of the behavioral profiles of these individuals then, or was that the idea?
Yeah, in a sense, yeah, I'm getting at the social environment that, you know, not just stability, but also the stability of the social environment. Where if you disrupt not just the numbers, but you disrupt sort of how similar they are to each other, that could potentially have an effect on the adult behavior and therefore, you know, hit the offspring in a way. I was wondering if you, I mean, I know these experiments were some time ago, so maybe this wasn't on the profile, that high profile at the time, but do you have any data on sort of the composition based on similarity in phenotypes?
No, not yet. That's an interesting question. I have never thought about that, I have to say. We have no data on that, but that would be very interesting, yeah. Maybe we could check it in our old data. Probably it is in there, but we have never checked it. But yeah.
Go ahead, I'm sorry.
Yeah.
Did you measure, do you know anything about the intrauterine environment that the offspring experience? So
no. Of course, that would be also interesting because of these studies of Münster, for example.
Yeah.
So the neighboring. So we know what we have done is that when we selected our offspring, we didn't use offspring only from pups where only one. Where no sibling was there. So they had at least one sibling. But of course, that could be brothers or sisters, and we don't know the position in the uteri. So, of course, it could be that, but that it makes no sense that the masculinized daughters are always near a brother, that they are more. Oh, wait, I have to think. What we have seen, that very old studies, we have once investigated sex ratio. And we could see that in the that the offspring of mothers who had lived in this unstable social environment, there were a sex ratio shaped in direction to the female. So there was a female-based sex ratio in an unstable social environment. So, but that is then the other way around. So that means that there are not more brothers, for example, there, because it was a sex ratio shaped in the female direction. But maybe I have really to check the old data. That's a very interesting point, whether we have something in there. But we have no really inter-uterine data. That was too complex.
Sure. Okay. Well, thank you very much.
Thank you, Lauren. I'm going to skip you, Eduardo, one sec. There's a student, Manuela, you can ask your question.
Hi, Manuela Cardona. I'm studying a master in University of Exeter. My question is if the individuals that used to live in pair that presented high aggressive relativeness, do you know if the pre-existence pair present like more affiliative interactions, like because we know that they present aggressiveness with when you put like the new individual, but what happens with the pre-existence pair?
I'm very sorry. It was very hard to follow. Maybe the connection or so.
I'm sorry.
Do you speak about the adolescence study? Or
The relativeness of the pairs when you had the colony and the pair groups.
Yeah.
I want to know what happened with the pre-existence pair and when you put the new individual in the group.
So you mean the other match-mismatch experiment, let's say. So if we have that we have a pair and a colony host male and put them now in an unknown colony. That's the idea? Okay. We have also done that experiment, and here that would represent a match for the colony host male and a mismatch for the pair host male. And we see that the colony host males have an advantage in the short term because they are less aggressive and they can be how is the English word? They can live in this group without stress, without aggression. And the pair host males, when they were put into this colony in the first 2 days, they have a high increase of cortisol and they are also highly aggressive. But after 2 days or so, they could reshape. That's also a very important point. They could reshape their behavioral profile and now they are also less aggressive. So it needs some time. So the first time is probably very costly for them, but they could reshape. And in this situation, we have also measured paternities again, but the colony host males and the pair host males were not able during that time we have investigated to reproduce. That were always the older males who could sire the offspring. So, but nevertheless, there seems to be in the short term advantage to have the correct behavioral profile, let's say, but it could be reshaped. That's always the case that the reshaping could occur, but probably it's costly.
Oh, okay. Thank you very much.
Thank you.
Thank you, Manuela. Eduardo, now your question.
Yes. Again, just trying to learn because we've done a few things trying to have similar approaches, but of course we have very, very different study design, very different number of subjects. What caught my attention is how small your measures of dispersion for most estimates of central tendency were. I mean, I don't know whether those are standard errors or standard deviations, but for example, in one of the last graphs where you were showing us the cortisol, the blood cortisol in males. So, and I don't know if I missed it, but roughly, what are the number of subjects groups that across so many years, so many experiments do you usually have? And is it a huge number? And that's how you end up with such small measures of variation? Because our experience, and this is feces, it's not blood. That's what I'm saying. Whole story. But the standard errors are huge. I mean, the variation within individuals sometimes is bigger than between, etc. So I just wanted to get into some of the details of your sampling and variation in the data.
Yeah. So normally the group size is normally 12 to 16 individuals per group. More is not possible because it's really a very long-lasting experiment and so. But 12 to 16 is the normal number of things. Of course, we have a high variation, but of the reactivity, the initial values are normally the variation is lower than in the reactivity. And what we also have investigated is stability in whether the individuals are stable over time regarding, for example, cortisol reactivity or also the basal values. And what it is interesting is that the basal values are not stable, but the reactivity values are stable. And that makes sense, I think, because of course, cortisol is a stress hormone, but nevertheless, cortisol is also a metabolic hormone. So it's needed to provide energy. And of course, during daily life, you need often energy. And then the environmental conditions are also changed in a short term, and the individual have to react. So it makes sense that the basal values, the initial values, are not stable because always different things are. But how to react to a stressor, this seems to be stable for each individual. And this we could show for the wild cavies, we could show also for the guinea pigs that this is the case. But nevertheless, so we see that there is a variation, but it's what's the English word? If we are doing the statistic, then we see nevertheless that the groups are significantly different. I think that shows that there is really a strong effect in cortisol reactivity. In this, I think at the moment that the cortisol reactivity is one of the best factor in mediating behavioral effects, I have the feeling, not initial values, but really the reactivity. And I also have often, so if I am allowed to add that, because you have said that you are measuring fecal corticosterone. Of course, we are doing that also in mice, for example. We could do that also in guinea pigs. We have validated that, but I have, in these studies, we don't do that because of several reasons. The first is that's not really non-invasive in our guinea pigs, because then to receive enough fecal sample, we have to put them out of her home enclosure and keep them also alone for 2 hours. Then they can also take a blood sample or so. And in the fecal samples, you not measure really the acute response. Right. So, of course, it's very good if you would compare enrichment or other long-lasting things or so. To measure really the acute response, that's not so good to use fecal samples. But it's a very nice tool, of course, to investigate several things. And at the moment, we are also establishing cortisol from hairs. That's also working very well. But then, of course, you measure against other things. And here it would be the best if you could, cut the hair, then start the experiment. And after 4 weeks, for example, you can then cut again the hair at this specific area of the body. And then you really know during this 4 weeks, for example, this cortisol concentrations has accumulated in the hair. Then you can really say something. The literature at the moment, if I read that, everything can be measured, of course. You will always measure in the lab cortisol, no problem at all. But how to interpret that, that's really not so easy. And for that, you have really to think it's better to use fecal samples, blood samples, saliva, hairs, feathers, or whatever. But I like to see what for possibilities are there, but we have to be cautious about that.
No, I'm totally with you. It's so much so that I think we need groups like yours, and maybe you have published this, but those who can really measure it in all kinds of substrates need to publish that and show us how what you measure in feces has no connection to what you're measuring in blood, because that's what we need to hear, those of us. I mean, we're stuck on certain things we cannot do only by blood, but I mean, we can only do feces, and that's okay. But what we cannot do is ignore the fact that our colleagues are telling us, listen, I can do it all. And let me tell you, in my system, when I do it all, the feces is a whole different story. So we shouldn't ignore that. So I encourage you, help us get this message across for those of us who are stuck with feces or urine or whatever, that it is not the same.
Yeah, exactly.
100% agree. Thank you so much. I do need to go now, but I'll see you Thursday.
Yeah, okay.
Thanks for the wonderful talk.
Yeah, bye. Thank you.
Thank you, Eduardo. Okay, I will ask my question. I already introduced myself. So my question is related to the effects of the unstable environments in the length of the pregnancy. And so if it does, I don't know if it makes it longer or it doesn't have any effect.
No.
And not at all. So
Not at all. No. No.
Interesting.
I was also surprised, but there was really no effect at all.
And then, the individuals, are they smaller? Do they have any like morphological difference with the?
No, also not. We know in mice, for example, could also show that also the masculinization effect of the daughters, and they showed that in mice you have this sex difference regarding the distance of the you know what I mean. So the distance between the anus and what is the English word about
the
that?
anogenital?
Yeah, exactly, the anogenital distance. And this is different between males and females. And so you can see is it a male or a female. And they could show that the daughters who have, that was not an instability, but there was a stressful event during pregnancy. And they showed that there is also masculinization effect, and they showed that they showed an anogenital distance as males. So there was also morphology and masculinization effect. In our guinea pigs, we don't see any morphological effects. So in guinea pigs, for example, males have a little bit broader head or whatever, so but they looks like females. You don't see it, and it's also very interesting. It's fun, I have to say. In a student course, I have videos from daughters of a stable or an unstable social environment, and since I think 50 years, I show them these videos. I don't say what's the sex ratio in this group. I say to them, now say, what do you think, how many males and females are there? Could you say what is the sex ratio in these groups? And then during the whole years, the most say in the groups with the daughters coming from the stable social environment, there was only females there. In the other groups, at least one male, but that's also only females. No student has had seen that correctly. They looks that's really interesting. And then we look at the behavior. What's the reason for that? They showed this masculinization. They behave as a as a male, but nevertheless the appearance is as a female. So I always astonished that they don't see that these are females, because they look like females, but they behave as males. But that's we don't see any morphological differences or so.
Oh, that's cool. But you mentioned also behavior. So they are more aggressive when they come from the unstable environment.
The daughters.
The daughters. But are
they are not aggressive. So they showed this male typical courtship behaviors. They showed also play behavior as males, because there's also sex difference in males. But we didn't see differences in aggression. That was in the males, but not in the females. Interestingly, I told you that we have also one study in which we applied ACTH. And if we are doing that, so that cortisol was increasing during pregnancy, these daughters were more aggressive. But they don't show masculinized behavior in the other parameters. So I don't understand it fully, I have to say. But that's there are different mechanisms seems to be in these different behavioral patterns. That is very complicated, it seems.
It does seem very complicated. Okay, I have more questions, but Suleima has a question as well, so I'm going to give her opportunity. And then
yes,
I ask more.
Suleima Tang Martinez. I'm retired from the University of Missouri in St. Louis, and I worked with rodents most of my life, a little bit of work with catamorphs. I was really intrigued by the fact that you found in the experiment where you looked at the gonadectomized versus non-gonadectomized, that gonadectomy resulted, if I remember correctly, in higher levels of cortisol and higher levels of aggression, and vice versa in the opposite case. And I'm wondering whether you have any thoughts about the mechanisms involved. Like, do you think that the cortisol is actually causing, quote unquote, the aggression? Or, I mean, usually we equate aggression and testosterone as being related. So whether this is a correlation that we're seeing or any other confounding effects, but why high cortisol, high aggression, low cortisol, low aggression? And before I stop, I just want to say I thought it was a fascinating talk, and thank you very much.
Thank you very much. So I think very important it is to distinguish between long-lasting increase of cortisol and aggression and the short increase of cortisol and aggression. Because in our case, we have only this short increase, and there are some studies in HAMS. Is hamster the English word also? In German, it's hamster. I'm not sure. Is it okay? That they could really show experimentally that if there is a short increase of cortisol, that this really has an effect on aggressive behavior. We have tried to show that also in our guinea pigs, but here the variation was too high. We have a trend, but not really significant effects that really this short increase or decrease of cortisol has these effects on aggression. But at least in hamsters, it has been shown. And so I think maybe that's often the case, that a lot of different factors can play a role. But I would assume that the cortisol reactivity at least plays one important role in mediating this aggressive phenotype.
Thank you so much. And the other point that I wanted to make is related to what Eduardo said right before he signed off. And it reminds me of a seminar we had by Lee Coren, I think it was last semester, in which she talked about the fact that the way that you measure hormones in fecal samples versus blood versus, other ways, that it makes an enormous difference. And I was really struck by that, because so often we assume that our, measures are accurate, and yet it seems to depend on the method that we use, what we get. So thank you so much.
Thank you.
Thank you, Sulema. And we have another question from Maren.
Hi. Sorry, I have another question that is not really related to your talk, but to the Münster KVs. It's another of my favorite anecdotes, and I'm not quite sure whether it is true or whether I'm misremembering actually what I saw, because I never found the publication. So when I'm talking about choice experiments in my lectures, I always tell the story of another of your experiments, that you had the guinea pig female in the middle, and then the typical choice experiment. So explain you to the others, you can do it better than I. I just want to hear whether it's true and whether it was ever published, actually.
So you mean this preference test? So the idea behind that was that we had wild cavies and the Gallia musteloides, the yellow-toothed cavies. And the interesting thing was that they have this very different mating system. So the wild cavie is polygyne, and the other is promisc. That was before we had these monasteriensis, that was then monogam, of course. And you can see the yellow-toothed cavy has really large testes. So it's really a promiscuous animal. So they need a lot of sperm to compete on sperm, on the level of the sperm, not on the behavior. But the idea was then that always it has been discussed that the male can monopolize females, like, for example, in the wild caves, or the level of sperm, there will be competition, but also always on the view of the male. And we wanted to know, does the female not also plays a role? And so we wanted to know female choice, let's say. And for that, we have conducted this preference test so that the female was in the middle of an enclosure, and around her, there were 4 different enclosures with males. For example, the wild cavies, there were 4 different cavies, and only the female has the possibility to visit the different males. The males couldn't go out to the female, but the females could visit the different males. And then we wanted to know preference test and also the social preference, but also whether they will mate with more than one male, or what is the role of the female. And here we could show that in the wild caves, which are showing this polygynous system, that the females are visiting more males, but they have a preferred male. And there are only, I think, 20% of multiple paternities. It could occur, but we see from the behavior that they visit males, but there are a preferred social partner and also mating partner. In the Gallia monasteriensis, they really want to visit only one male, so it's really monogam. And in the Gallia musteloides, the promiscuous female species, the females are really went to each male and they mate with more or less all of these males. And so that's really much different, depends on the mating system. And interestingly, regarding the Gallia musteloides, the promiscuous species, the multiple paternities are, I think, about 80% or so. That's really a lot. And we could also show experimentally that the offspring coming from a litter with more than one father have a higher probability to survive. We have only investigated this until the age, I have no idea, I think the first 100 days or so, but we had more litter who was born dead, or before weaning maybe they were dead. That was more often when there was only one father. So there should be also maybe an advantage to mate with more. That's often discussed, of course, that there is really an advantage for survival and for fitness if the females are mating with more than one male in this species. But that was the experiments you remember?
Partly. What I meant was this the traditional setup of the study design, where the female just sits in front of the males, and that there was measured how long do they sit in front of them.
Okay,
and then you opened it up, and then they could go to the male. And I always tell that they were beating up the male that they had been sitting in front of, because that's what I saw on the poster, but that's what I want to confirm or not to confirm.
Yeah, no, that's true. They are sitting before, then we have measured really, because the idea behind that was that we wanted to distinguish between social preference and mating preference, because that's sometimes not the same. And that was the reason that we have started only with sitting before. So there was a grid or so that they can sniff each other and of course hear each other. And then, to be honest, I could not really remember the results correctly, but if I remember correctly, the wild cavies are really preferring that, what I mentioned before, that they really preferred one male also already in this phase of this experiment. But maybe I'm wrong. It's really long. I think it was 2002 or so. I have no idea. I have to check it out. But you're completely right. We had these two different parts of these experiments. Does that now answer your question?
Yeah, I think so. So what I remember was that essentially they, in all three species actually, and it was, I think, perhaps just on a poster, that in all three cases they switched the preference so that they were sitting in front of one male, and then you opened it up and they went in, beat the male up, went out again, and then went to the other male and mated with the other male.
They
that's at least the anecdote that I always told the students to tell them, be careful with traditional setup of mate experiments.
Yeah, I remember that they have or visit all males, but then I'm pretty sure that in the wild caves they still then preferred one male, but I'm not remembering whether it's the first male or another one when they have then visited all males. If I remember correctly, the preferred mating partner was always the largest male. So the body weight was very important. So they preferred for the mating the largest male, but not for the social preferred partner, if I remember correctly. But please, I'm not sure about that completely. But that they choose the largest male for mating, I'm relatively sure, let's say. The other, I'm not completely sure. I'm sorry.
Interesting. Thank you, Maren. I'll ask a question, and then there is Suleyma's question as well. And I'm just going to ask it because it's a follow-up question of Maren's. And I want to know exactly, like, how do you measure or make a difference between the social preference and the mating preference? Like, what kind of behaviors do you actually measure for this test?
So the social preference was, for example, how often they were nearby the male, and that they show sociopositive behavior, for example. And the mating preference is paternity, so that they are really mating, copulating with the male, and is he then the father? Or maybe she is visiting more males, mating with more males, and is then there are also multiple paternities there. But that's the behavioral pattern is really the what is the English word? The nearness, closeness to the male,
or proximity.
Proximity, exactly.
But not contact, or just close proximity, or contact.
Contact, it depends on the part of the, when we have this grid between the animals, then they have not really the possibility to contact really, but they can sniff each other or so. So each social interaction which is not aggressive, that's what I would say, is really this a sign for this social preference. And if we would, we also have studies regarding bondings or so. If we have bonds between males and females in guinea pigs, it's a little bit easier because here they have really behavioral pattern. We have talked about this rumping, flanken in Germany. I am not sure. They do that only with a preferred female. And this is a very clear behavioral pattern for a close bond between the animals. I'm not aware about this is also true in wild caves, I have to say, but probably maybe also, but I cannot remember that we have measured this behavior in this preference test. I have to look it up, but that of course would only be possible when they can visit really the male, go into that. But here we have sociopositive behaviors and how close they are. That's the most important indicators.
And the behavior that you mentioned, that it's a characteristic of the bond, flanking. So it's the I don't record. So can you describe it to me? Because I don't. Probably we have it as well, but I don't recall the name of it.
That the male is more or less jumping over the female and pressing the gland, what is the perineal gland, press it on the body of the female. And so it's also marking it more or less, but it's only doing with his preferred female. It's interesting. I think it's a behavior which is really very clear, but nevertheless, in a student course, for example, when they have the first time they have to describe the behavior, to do an isogram or so, nobody is seeing the flanking, there's a rumping. They don't see it. And then they show them a video clip. Here, now, oh, we have seen that. We have not recognized it. I don't understand that because it's really not so, you can see it. And, but it's, maybe it's also only shown in, in a reasonable amount, let's say, in mixed sex groups. If they are living in pairs, then it's probably not necessary, I don't know. Then you see it only very rarely. Whereas in the mixed sex group, for example, in the colonies, you see that very clear, and then it's really a good indicator for the bonding. And also rumba, it's English, rumba or rumba? I was not sure about that.
I don't know, but I say rumba.
Okay, rumba. You can also see that this rumba is also, the frequencies of rumba is much higher to the bonded female partner than to other also known female. They are around him, but he don't show this behavior if there is no bond between these animals. So that's also a good indicator for that. But that's also always for domestic guinea pigs. We have not really investigated this in detail for the wild caves. There we have really only these preference tests, which Marvin has told to us, and it's really long ago.
Okay, thank you, Sylvia. And Suleima?
Yeah, I was really interested in the last two questions and discussion, because we actually in my lab did a study using very much the same type of setup, where we were looking at preference versus mate choice. And in our case, we were looking at preference and mate choice, where a female had to choose between a full sib, a half sib, a cousin, or a totally unrelated male. This was in prairie voles. And we also, if I remember correctly, it was a long time ago, and I never published it. I'm thinking I should really sit down and publish it, or try to publish it. It's difficult because we had very, very strong trends in what we saw, but because of circumstances we couldn't control, we weren't able to test more to, try to get a large enough sample size, but very, very strong trends. But at any rate, it was fascinating because it was the same sort of thing. Generally, whether male or female subjects, they would go in and they would enter the box that had the full sib, and they would just go in there and spend enormous amounts of time, cuddle, like literally sit, touching, cuddling with the sibling. But then they would, after some time, they might leave and wander around and then mate with one of either the cousin or the unrelated male most frequently. But I think it's a really, really important distinction that needs to be made. And so many studies, particularly in the past, were really looking at social preference and not at mating. And I know I've been a stickler on that, and said at times, well, this shows there's a social preference. This does not show that there's a mating preference. And it's just interesting to see that, you had the same sorts of things. And we did the same thing. We measured the amount of time spent in a compartment and also with a potential mate, and also any physical contact. I'm not sure if we did just proximity because our voles were so, touchy-feely that they would just clump together. But yeah, there's an enormous difference, and I was just fascinated that your results were the same as we saw also. So
if you publish your data, I would be very interested in it.
Well, I could even make it. I have to find it somewhere in my office, but I could send you what we have.
Yeah,
and
that would be great.
I think the problem is so difficult to publish data that are not statistically significant, even though, especially if you look at SIV, full SIV, full SIV, half SIV versus cousin and unrelated. That's where you see the biggest difference. And it's very close to significance, but it's not. So, yeah.
Yeah. I hope this is changing, because a lot of journals now really say that also not a significant difference, of course, are important and should be published.
Of course.
And that's, I hope so, that will become more and more, because it's also so important in animal experiments, because we have to think about how many experiments we are doing with animals. And if we don't know that other people have done the same, but couldn't find differences, we do again the same, and we also don't find differences, that from an ethical point of view, it's really a very big problem, I think.
Yeah. And then there's a whole issue of what statistical significance really means, which is something that in this group, we have discussed extensively. But so that's the system we're working under, unfortunately.
Yeah, true.
Okay, thanks. I just wanted to throw that out because I was really struck by the parallels, both in the methodology. It sounds like we had our voles tethered so they couldn't leave their compartment except for the subject who could visit everybody else. And also the similarity in the results. It's reassuring.
Yeah.
Thanks so much.
Thank you.
Thank you, Suleiman. Your setup is the same as the setup I use, with the focal just running around and the other ones just tied up to the to the endings. Lauren?
Yeah, I just have a follow-up to that. If Suleiman is still there, I think one thing we can do is actually report those non-statistically significant results and emphasize the effect size. And then what I like to see in those situations is, since you have comparisons with the cavies, you could have a table of species with similar trends or different trends to give it a sense that this is a bigger picture. And I think that's a way to get around this bias towards p-values being low. Send it to JMAM. Got 2 editors here, right? I mean, for JMAM, we'll look at it.
Yeah, you may be getting 2 very, very old studies that I never got around to publishing at some point, but I have a big paper that I have to finish first. So,
well, at least one of us will be there, I hope, for another few years.
Yeah.
I don't know how much longer I can go with it.
Thanks.
Okay. Thank you, Suleiman. Thank you, Lauren. And so I'm going to just take advantage, and I think I will close the seminar for today. I want to thank Sylvia for her incredible talk and amazing discussion. Thank you everyone for attending, and I remind everyone who's still watching that we have seminar next week. So we're going to be talking about amphibians and parental care. So everyone is welcome. And now I'm going to...
I would also like to thank, it was really nice to give this talk here. I liked that a lot. And also the discussion was really very nice. I like this too. So thanks a lot to all of you. And I'm sorry that I had to postpone the talk from May to October, but that was, yeah, I couldn't change that. But I was really happy to be here. Thank you.
No, thank you for coming. Well, thanks everyone, and we'll